So there's two sources of fat, and I know you know what they are. Looking at the pictures or reading my description, who could tell me which is which? What's first? Comes mainly from animal sources such as meat, eggs. Well, I mean, that's involved there. The two fats, one describes more liquidy and the other is more solid.
Unsaturated and unsaturated.
Which one?
Unsaturated is from the plant and fish sources, and saturated is from the meat.
Good job. Whoops. And obviously, this is a guide because there's some nutritional portion to bio on switching from unsaturated to saturated. Yes. Oh, okay. But remember all that little font you have. You're only looking for the two words that I just described, which was saturated and unsaturated. Hi, Miss Calder. Let me pause. You did?
He gave this to me, and I forgot. But it's not his fault. He did give it to me. Oh, it's like that too? Yeah. What was the due date?
No, the thing is
He gave it to me Wednesday.
the due date was Wednesday.
Okay.
However, let's start with composition. It should be a little bit easier now. Who could tell me the composition of a protein? Okay.
Carbon, hydrogen, and nitrogen.
Correct. You're adding one element different from carbs and lipids, which is nitrogen. It's a combination of carbon, hydrogen, oxygen, and nitrogen. What is the building blocks? Look at my little picture and it'll kind of remind you. What's the building blocks of protein? Amino acids, good. Now, every amino acid is attached to what? Look at the picture again. I'll make it bigger.
Carboxyl.
Good. A carboxyl group and all you're doing is interpreting diagrams. I mean, the amino group is the amino acid. It's attached to a carboxyl group and a what?
Side chain.
Side chain. R. Good. Now, what's the function of a protein? Here's where I get a lot of answers that kind of fall into the other two major organic compounds. When you think of a protein, what automatically comes to your mind? Muscle building. Good. That's exactly what it's for. It's kind of responsible for almost all your physical characteristics. So muscle building, your leanness, your physical characteristics. What else is the function of a protein? And I'm going to have you add that to your notes. Not energy. Because we know what's responsible for energy.
Lipids and fats.
Lipids and fats. Which one is long-term energy?
Fats.
Fats and lipids. What else is energy? Carbs. And what do? Quick, fast energy. Okay, you want to store the energy, it's fats. So what else do proteins do besides physical characteristics? They form what?
Muscles.
Muscles, yeah, we said that. That's part of physical characteristics. I'll show you. They form hormones, okay, which actually aid in this. And they also form enzymes. Remember enzymes? What is an enzyme? Who could tell me?
They speed up chemical reactions.
Perfect. They are catalysts. And I know that's kind of small, so I'll make it bigger, which means they speed up chemical reactions. Now, every enzyme, have you heard of the lock and key explanation? What does that mean? Does anybody know? What's a lock and a key?
It's for that it would be maybe a place where it goes and it only fits one type.
It's like a perfect fit. And for every enzyme, or for every chemical reaction, there's a perfect enzyme that's going to fit and help speed that reaction. Without enzymes, our body would not be able to break things down at the pace it does, or even make things at the pace it does. Every reaction that we learn in this class, an enzyme guides the reaction. So it helps it move along. And I guess I have some animations, which I could probably just show you on Monday or Tuesday before the review. I guess I could show you this one. I'll come back. So here's an explanation of how enzymes work.
Speed up chemical reactions in the cell. A special feature on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex.
Basically what it's trying to tell you there is when the enzyme attaches to the molecule, it actually did what? What did it do right then when it attached? Formed a what? A bond and a compound in its own, okay? It's another combination of atoms that's going to speed up this reaction. And it's a perfect fit so that whatever it was looking for each other. So, because molecules are always moving, they're always trying to find something.
Actions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These encourage between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyzed the formation of two products from a single substrate.
Okay, I know that ending was like, all right, how can I go back now, is the question.